Well, I first got the idea of oral roots from a discussion with Indigenous people about oral histories and the passing of traditional and cultural information from mothers to their daughters about the healing properties of plants. And then I became an artist in residence to learn about the neuroscience of audiology, hosted by Symbiotica at the University of Western Australia. I was also influenced by my visits to the hospital at the University of Zurich, where they work with hearing-challenged babies, children and adults. The result was Aural Roots, an interactive installation that encompasses 3 embodied sonic environments based on these experiences. It offers a metaphorical learning experience that scales down the viewer to 5 nanometers and places them inside the organ of Corti in the inner ear. Here they can trigger acoustic reactions by manipulating sculptural models of moving hair cells called the stereocilia. Each stereocilia triggers sound compositions based on pitch or frequency. As a resident artist in Symbiotica, I spent all of my time in audiology. Here I learned a lot about the physiology and anatomy of hearing and how important it is for learning.
The philosophy of Symbiotica is to enable artists and people who are not usually engaged with the life sciences entry to the lab so they can experience what it means to manipulate life. And the reason for that is because we believe that we're lacking cultural language to engage with what's going on with life at the moment within the context of science and engineering.
My second residency was at the Laboratory of Experimental Audiology at the University Hospital in Zurich, Switzerland, where I learned about hearing loss with patients. Here, I learned about the functions of the organ of Corti. The organ of Corti sits inside the cochlea in a snail-like structure. It is lined with a set of outer and inner moving hair cells called the stereocilia that move according to the waves of fluid that are triggered by the eardrum. When the stereocilia are moved, potassium travels up them to their cells and onto the auditory nerve and through various centers like the midbrain and finally to the auditory cortex of the brain itself.
The hair cells are specifically designed or have evolved to respond electrically to minute vibrations that are caused by sound. Those electrical responses are then transmitted to the afferent nerve fibres, and that information is then sent to the first stages of the brain, the cochlear nucleus, and from the cochlear nucleus to the olivary complex and to the inferior colliculus. Those electrical signals are then relayed further after processing, further up to the medial geniculate nucleus, part of the thalamus, and finally to the auditory cortex, where conscious perception takes place.
Without our stereocilia, we cannot hear anything, and they are unable to regenerate. For example, just in one of my ears, I have about 3,500 inner hair cells and about 12,000 outer hair cells. The inner hair cells relay harmonic information to the brain through the auditory nerves, while my outer hair cells work to amplify the sounds and recognize the pitch. This is the diagram of the cochlea. While the low-pitched sounds coming into my ear affect the stereocilia in the smaller end of this spiral, the mid-pitch that I'm hearing occurs exactly in the middle. High-pitched sounds are registered in the larger entrance of the snail-like structure, and therefore most damage to our hearing occurs within the high-pitch range.
What happens is the hair cells in your cochlea, which actually kind of catch the sound wave, so they follow the sound wave, and the movement of the sound wave moves the stereocilia, and that causes eventually the process whereby you get electrical activity in the brain. So these stereocilia is extremely important for hearing. As soon as you have damage to your stereocilia, if you wouldn't have any stereocilia, you're deaf. Because the cochlea is connected to the brain via the auditory nerve. So the auditory nerve are the nerve fibers that come from the inner hair cells. And they project to the cochlear nucleus, which sits here in the brainstem over there. So that's the first relay station of information. And when I say relay station, I mean that that's where, you know, the first processing takes place of the information coming from the cochlea.
Inferior colliculus is a really important relay station in the whole auditory nervous system. Everything that comes from the cochlea, both left and right sides, goes through brainstem centres but eventually has to pass through the inferior colliculus. It's the gateway to the cortex, if you like. So if we understand what's going on in the inferior colliculus, the auditory midbrain, then we have more of a handle on the information that's going to higher centres.
I learned that the activity and response of the nerves can best be measured in the inferior colliculus of the guinea pig. But first, I had to learn more about the pathways of efferent and afferent nerves that carry signals from the ear to the brain and back again.
Efferent input, we say, is going up towards the cortex. Efferent input comes from the cortex and goes down. And then the particular efferent fibers in the cochlea that project to the inner and the outer hair cells, they originate in the brainstem.
Well, because I'm an artist, I have always had to draw in order to understand this anatomy and where the malfunctions might occur.
Ever since we started research in this lab, we've been determined to look for applications of the basic physiology that we've been investigating. I think the fundamental belief there is that if you understand the normal physiology of a system, you're in a much better position to understand pathology. And there's some very clear examples of that in our research. So for instance, the study of the fundamental biophysics of inner ear vibration and the role of that outer hair cell active electromechanical motor, that now underpins all our modern day understanding of sensory neural hearing loss. And it also has generated spin-off in terms of a variety of diagnostic devices.
I asked Don to explain to me how this electrical activity from sound is measured in the auditory lab environment.
The basic starting point in any experiment like this is to have an experimental animal, and the animal we use is the guinea pig. The guinea pig is fully anaesthetised, so it experiences no pain during these actual experiments. But the guinea pig is located in this soundproof room here. We record the activity of nerve cells in there, and that activity is then piped out to here and displayed on the various recording apparatus here.
Actually, this experience of learning how to use this tool on real animals and in real time contributed a great deal to the design, sound samples, and construction of the final artwork. But one of the main inspirations for the form of the stereocilia came from images I found in the scanning electron microscope. Just look at the texture and shape of such motile cells. Here you can see the clusters of the cells at the scale of 5 nanometers. So along with a group of my close friends in the studio, we covered a set of wooden poles with small pellets of beeswax so that the surfaces resemble those seen in the motile cells of the stereocilia on this scale. The final resultant surface is moulded with plaster. Then these moulds are filled with fiberglass. And the outer and inner hair cells of the stereocilia were all produced in this way. Then the test began on the hanging apparatus of the set of outer and inner stereocilia hair cells, and wires were then mounted into them for the hanging mechanism. The stereocilia swing on balls that are connected to joysticks, and they were filled with foam, so they like to move and painted white. They are also tip-linked together to affect the other hair cell movement, and that's what happens in real stereocilia. The 18 stereocilia are then hung on scaffolding in 2 bunches, representing the outer cells, based on harmonics, and the inner cells based on volume. The touchscreen was then added to the back of the whole structure to show the organ of Corti and 3 different pitch ranges. This is where the programming starts. Together with 2 programmers, one on Max MSP and another on C++, numbers were sent from joystick positions to a program on a Mini-Mac computer. The viewer can touch the screen to travel up and down through the cochlea, from the high pitch environment to the media pitch environment and down to the lower pitch environment. There are 18 tracks available for the viewer to make compositions in each environment: low pitch, mid pitch, and high pitch. The womb is a watery, muffled environment. The embryo can hear sounds from inside the body, like the mother's digestion, her lungs breathing in and out, or the blood cells rushing through her veins. If the viewer plays with the outer stereocilia, they will hear sounds from outside the mother's body, like traffic noises, a rushing river, or even a refrigerator motor. It has been proven that sound is the first sense to develop in the embryonic state. Here the embryo can differentiate between sounds from inside and sounds from outside the mother's body. These low-pitched sounds are all based on real recordings from a collaboration with women in the university hospital who agreed to have contact microphones placed in their wombs. Speech actually exists in the mid-range of our hearing frequencies. Many communities practice oral storytelling, the passing of knowledge from one generation to the next. Here in Oral Roots, oral stories are told by Australian Indigenous women to their daughters about how to gather plants and roots for survival and medicine. Due to colonisation, this knowledge is unfortunately rapidly disappearing. The stories in the mid-range composition were made with Indigenous Aboriginal Australian advisors at the Koori radio station in Sydney. They're based on talks, interviews and written information by community elders and Indigenous researchers about the healing properties of plants. On the outer stereocilia hair cells, the viewers can relate these sounds to landscapes where the plants are found out in the country.
In the old days, the old people, they would be strong and happy. There was plenty for everybody and everything shared. Now we go to the shop. Yam is important. We have the one you call long yam. In my country, the old people would dig, the children would watch.
This composition is designed to focus on the high pitch range, because this range is normally the first hearing loss problem in older age. I gathered these sounds from the hospitals and the lab environments. For example, the sound samples here were recorded together with Professor Mulders and represent the firing of neurons in the midbrain of animals. Here I became a kind of sound composer myself. By interacting with Oral Roots, the viewers also become sound composers.
So what we're trying to do is find,
Try to find the frequency that this neuron is most sensitive to.
right.
Because we are recording in the midbrain of the guinea pig. And so these neurons are in the auditory system, so they're sensitive to sounds. So we present sounds, and you can actually manipulate the different frequencies, the different pitch. And you can also modulate the intensity. And you look for the frequency, so the pitch to which this neuron is sending electrical signals most vigorously. So it's spiking most vigorously.
In my visits to the audiology lab at the University of Zurich, I also recorded their tests with hearing challenged people and used these in the high-pitch composition.
My main reason to be interested into the changes that take place after deafness is because we think that some of these changes are associated with tinnitus. So tinnitus is a phenomenon of having a phantom hearing sensation. People often describe it as a hissing or buzzing or a ringing in the ears. We know that the brain has different changes when you are turning deaf. So I'm looking at the changes in electrical activity in the brain. When you are exposed to loud noises and you don't use ear protection, that this can then or either lead to immediate hearing loss, or it can actually accelerate your age-related hearing loss.
When you talk about hearing loss, there is of course a degree of hearing loss which you can determine or which you are talking about. If you have only a moderate hearing loss, you can still understand speech, hear all the sounds more or less. A few sounds are a little bit damped. But if the hearing loss is more profound, you start to lose some perception of special sounds, like you don't hear the birds anymore, you don't hear people who are whispering, and especially you don't differentiate between different speech sounds. And if you are profoundly deaf, then you may feel some vibrations, but you don't hear the sounds anymore. You cannot rely on spoken language for communication.
I learned that damage to the stereocilia is one of the most common forms of hearing loss today. At this lab, they are well known for their cochlear implants that can be placed directly into the cochlea to substitute the stereocilia.
You see the ear with the external ear canal, the middle ear and the cochlea. And here is the implant case below the skin. And we can have a look at how the implant looks like. And this is a real implant demo, which is put on the bone and then has to be connected to the inner ear with this electrode carrier. Some signals from the outside, so that's the speech processor which is being put behind the ear, and the antenna which transmits the signals through the skin into the implant.
There are many scientists working on hearing loss, and the condition of the stereocilia can be seen in these histologies. One potential research lies in the regeneration of stereocilia hair cells from supporting cells. But this research is only in the beginning. Meanwhile, hearing loss is an increasing problem in our society.
I think this artistic project could have important implications for hearing science, in fact. Hearing is an absolutely vital sense, and loss of hearing has devastating consequences for adults, but also for children. If there's any hearing impairment, then learning, intellectual development, all these things are impaired. So from my perspective as a scientist, anything that improves the public's engagement with hearing science and helps them to understand or to want to understand more about the fundamental physiology of hearing could have really important spin-offs in terms of awareness of hearing problems and a drive for cures of particular conditions.
Her work here is to deal with mainly one specific issue within problems of hearing loss, and that's the loss of those cells in the ear that have the hairs that allow us to hear. So those cells can't regenerate, so once you lose them forever. And her work is to try and recreate the experience of having cells that are connected to the cochlea, those hair cells, within a larger scale. So the audience can actually come into the museum, the gallery, and actually experience what those cells are and how they work.
Well, the bush onion, or yaranyari, is a grass-like plant that likes sandy soils and only grows in the dry parts. My mother used to find it on the beach dunes or in the sandhills near water. It has many small roots that make a very tasty snack. They are easy to dig up with your hands in the loose sand. The shoots come up after the wet season rain, but then you must not eat the roots, because that's when they're growing.